Right now, take a breath in through your nose. Before you exhale, take a quick, smaller sip of air on top of it, just past the point where your lungs feel full. Now let it go slowly through your mouth, longer than feels natural. That is a physiological sigh. You have done it many times without noticing, usually right before you cried or right after you stopped. What you just did on purpose is something your brainstem does automatically about every 5 minutes for the rest of your life. Understanding why it does this will explain why you feel calmer than any amount of just relax ever could. Start with the plumbing. Your lungs are not a single balloon. They contain around 480 million alveoli, tiny sacs at the end of the airway tree. Each one is wrapped in capillaries and is responsible for small parts of the total gas exchange surface, which, if spread flat, is about 70 square meters, roughly the size of a badminton court. This area is folded into a space that fits under your ribs. The only reason those sacs don't collapse after every exhale is a substance called pulmonary surfactant. This thin film of phospholipids lowers surface tension similarly to how dish soap keeps a bubble from popping. However, surfactant is not perfect, and neither is your breathing. During normal quiet breathing, which physiologists call tidal breathing, you only use a fraction of your total lung volume. Small clusters of alveoli, especially near the bottom of the lungs, slowly collapse. This is called atelectasis, and it is happening in you right now, in a small, unremarkable way. It has been occurring since you were born. Here's a part most people never hear. Your lungs have a built-in fix for this, and it activates whether you think about it or not. About every 5 minutes, without any instruction from you, your brainstem forces a breath that's nearly double the volume of a normal one. This is the spontaneous sigh, not the emotional exhale you associate with frustration, but a silent, automatic, mechanical event that happens 9 to 12 times an hour in a resting adult. Its job is to reopen those collapsed alveoli, restore surfactant distribution, and keep the exchange surface at full capacity. If you miss enough of these, while under anesthesia, on a ventilator, or in certain neurological conditions, lung compliance drops, oxygen levels fall, and the tissue starts to stiffen. The sigh is not about mood, it's maintenance. Anesthesiologists and critical care doctors actually incorporate a version of this into machines that breathe for patients who can't breathe for themselves. Standard ventilator protocols include something called a recruitment maneuver, a periodic, deliberately oversized breath, sometimes paired with a sustained higher airway pressure, delivered occasionally to reopen collapsed alveolar units before they stay collapsed long enough to cause lasting damage. CPAP machines, used for sleep apnea, work on a similar principle, holding constant positive pressure so those units never fully collapse. In other words, engineers spent decades recreating in steel and silicone a solution your brainstem has already been using since before you could form a memory. The difference is that a ventilator's recruitment breath is timed by a technician watching a monitor. Yours is timed by a cluster of a few hundred cells that never went to medical school and never needed to. It's worth considering why the reflex is set up as 2 inhales rather than 1 deep breath, because the answer is based on physics. A collapsed alveolus behaves according to Laplace law, the same principle that governs soap bubbles. The pressure needed to reinflate a sphere is inversely proportional to its radius. This means the smallest, most collapsed alveoli require disproportionately more pressure to reopen than partially inflated ones. A single large inhale raises pressure smoothly and can plateau before the most stubborn, fully collapsed units reach their opening threshold. The second, stacked inhale, coming while the lung is already near peak volume, with the chest wall stretched and intrathoracic pressure elevated, delivers an added pressure spike on top of an already high baseline. This is exactly the kind of extra push those high-resistance alveoli need. It's less like blowing up one balloon and more like giving a stuck drawer a second. Sharper tug right after the first one only got it partway open. Mechanically, the second inhale does different work than the first. But knowing that a sigh reinflates alveoli doesn't explain why doing 2 inhales on purpose, followed by a long exhale, makes you feel like someone loosened a strap around your chest within 30 seconds. For that, you need to look beyond the lung tissue and into a group of neurons in the brainstem that nobody clearly mapped until surprisingly recently. In 2016, Mark Krasnow's lab at Stanford, along with Jack Feldman's lab at UCLA, published a paper in Nature that mapped the actual circuit. They found a small group of neurons, about 200, located in a breathing control area called the retrotrapezoid nucleus, or RTN. These neurons release two neuropeptides, neuromedin B and gastrin-releasing peptide, both part of the bombesin family, which is also found in the skin of certain frogs. The RTN neurons communicate with the pre-Botzinger complex, the actual pacemaker of breathing that generates every normal breath you take without any conscious effort. When neuromedin B or gastrin-releasing peptide reaches the pre-Botzinger complex, it doesn't just add extra breaths. It changes a single breath into the double inhale sigh pattern. The researchers then conducted an experiment that made the mechanism clear. They used a targeted toxin, bombesin conjugated to saporin, to selectively kill the neurons that express neuropeptide receptors in the pre-Botzinger complex of rats. Before the injection, those animals sighed about 24 times an hour. 3 days later, sighing dropped to around 5 times an hour, a reduction of nearly 80%. The rats continued to breathe normally. Ordinary tidal breathing remained unaffected. What disappeared, almost entirely, was the sigh, and only the sigh, isolated by targeting a few hundred cells out of the entire brainstem. This is as close as neuroscience gets to isolating a single behavior down to its wiring diagram. Somewhere in you, right now, about 200 neurons are quietly deciding when your next big breath happens, and you can take control of that decision by choosing to sigh intentionally. The same pre-Botzinger complex that generates the sigh is also the circuit that opioid drugs suppress, which is how fatal opioid overdose occurs. Opioid receptors are densely packed on pre-Botzinger neurons. At high enough doses, opioids not only slow breathing, they flatten the entire rhythm generator, sighs included, until the maintenance of reinflation stops and the urge to breathe fades away. It's a sobering detail in an otherwise soothing story. The same few hundred cells that provide you with a quick, free, physiological reset are also, when chemically silenced, the reason people stop breathing in an overdose. One circuit can lead to two very different outcomes, depending on whether you are activating it or suppressing it. The story shifts here because the alveoli and the brainstem circuit only explain half of what you felt 30 seconds into that first sigh. The other half involves a completely different system, the one that controls your heart rate. Running through your chest is the vagus nerve, the longest cranial nerve in the body. It travels from your brainstem through your neck, heart, lungs, and into your gut. It is the main pathway for your parasympathetic nervous system, the rest and digest branch that opposes the sympathetic fight-or-flight response. Every time you exhale, vagal tone to the heart increases slightly, causing the heart rate to drop. Every time you inhale, vagal tone decreases slightly, and heart rate rises. This pattern, known as respiratory sinus arrhythmia, is measurable on an EKG in anyone. It's why slow, exhale heavy breathing calms you while fast, inhale heavy breathing, similar to what you do when you're panicking, makes you anxious. A physiological sigh is designed to be exhale dominant. The two inhales are quick and almost clipped. The exhale that follows is long, often lasting 5 or 6 seconds or even longer. This extended exhale is a deliberate pulse of vagal activation sent straight to the sinoatrial node of your heart, the small patch of pacemaker tissue in the right atrium that sets your resting heart rate by firing its own spontaneous electrical rhythm about once a second. Acetylcholine released by vagal fibers onto that node slows the firing rate directly, similar to how easing off a metronome slows a beat. This is why your heart rate noticeably slows within 1 or 2 cycles of physiological sighing. It happens immediately, not after you calm down. Clinicians who study heart rate variability, which is the fluctuation between heartbeats, use this exhale-driven vagal signature to indicate a healthy, flexible autonomic nervous system. Low variability, where the heart beats almost uniformly under stress, is linked to worse cardiovascular outcomes and higher levels of anxiety. A long, sighing exhale is one of the quickest ways to manually increase that variability in real time. There's a third layer at play that is chemical, not mechanical. Your brainstem not only monitors how much air is in your lungs but also checks the partial pressure of carbon dioxide in your blood, primarily through chemoreceptors that respond to pH changes in the cerebrospinal fluid. When you're anxious, you tend to breathe shallowly and quickly, using chest muscles instead of the diaphragm. This oddly causes you to expel too much CO2 compared to what your body needs. The drop in CO2 raises blood pH, creating a state known as respiratory alkalosis, which brings its own symptoms: lightheadedness, tingling in the fingers, and a specific sense of dread unrelated to your original concerns. It's the way you breathe that generates anxiety symptoms, not anxiety causing a change in breathing. This causal relationship is often overlooked. The double inhale sigh, which maximally recruits alveoli on the way in and slows the exhale on the way out, helps restore CO2 to a stable level instead of further depleting it. This contributes to calming you rather than agitating you, unlike rapid breathing techniques such as Wim Hof-style hyperventilation, which do the opposite. The sigh reflex continues even when you're unconscious. Sleep researchers, including those from Jan Marino Ramirez's lab at Seattle Children's, have studied sighing across sleep stages, finding it clusters around transitions. It occurs when shifting from light sleep to deep sleep, during brief awakenings throughout the night, and when entering or exiting REM sleep. The same prebotsinger circuitry that reinflates your lungs while you're awake also works quietly between sleep stages you won't remember crossing. This is why researchers now view sighing less as a specific reflex and more as a general marker the brainstem uses whenever a physiological state is about to shift, from awake to asleep, calm to stressed, and back to calm. In a very real sense, you are utilizing the same reset mechanism at 2 p.m. under pressure at work and at 2 a.m. while sleeping. But a fourth mechanism operates simultaneously, and it has nothing to do with gas exchange. It relates to pattern. A research group at the University of Leuven, led by psychophysiologist Vlemincx, spent nearly a decade studying something few had tried to quantify. Not how deep or how fast people breathe, but how varied their breathing is from one breath to the next. Using a wearable respiratory monitor, Vlemincx's team measured the timing and volume of each breath in resting participants. They found that healthy breathing isn't perfectly regular or chaotic. It exists within a specific range of structured variability, with breaths related to their neighbors but not identical. Under ongoing mental or emotional stress, that structure breaks down. Breathing becomes either too rigid, set in a narrow, effortful rhythm, or too erratic, varying unpredictably. Vlemincx discovered that both patterns are followed by increased spontaneous sighing. She proposed what she called the resetter hypothesis. The sigh not only refills alveoli but also interrupts a destabilized breathing pattern, pushing the entire system back into its normal operating range. This is akin to resetting a piece of electronics that has started glitching by cutting the power and rebooting. In a 2010 study published in Physiology and Behavior, participants who sighed, whether spontaneously or by instruction, reported significant relief afterward, and their breathing variability shifted back toward a healthier range. In this view, the sigh acts less like a valve releasing pressure and more like a reboot. There's an interesting, almost tender piece of evidence supporting this from outside human research. In a 2005 study, physiologists Adam Soltysik and Adam Jelen exposed rats to a cue predicting an upcoming shock. Sometimes, a second cue signaled that the shock had been cancelled, indicating safety. Sighing in the rats increased about 20-fold right after this safety signal, far beyond levels seen during the threat or normal rest. The sigh didn't indicate distress. It marked the exact moment distress ended. Other rats in the same room, just observing, started sighing too. This behavior seemed to spread socially, as if one sigh signaled to others that danger had passed. It's a small but intriguing observation that aligns with a common human experience. You usually sigh not at the peak of a bad feeling but in the exhale right after it resolves. Now think about what happens when all four of these systems, the mechanical reinflation of alveoli, the peptide-driven brainstem circuit, the vagally mediated slowing of the heart, and the resetting of unstable breathing variability, activate simultaneously, on command, because you chose to breathe in a specific way. This convergence is what a team at Stanford Medicine sought to measure directly in awake, stressed humans, not just in rats. In January 2023, Yilmaz Balban, working alongside David Spiegel and Andrew Huberman, published a randomized controlled trial in Cell Reports Medicine. They recruited 111 volunteers and divided them into 4 groups, each assigned a different 5-minute daily breathing practice for 28 days. One group practiced cyclic sighing, repeated physiological sighs involving a double inhale followed by a long exhale for 5 minutes. Another used box breathing, a military and clinical method that involves equal durations of inhale, hold, exhale, and hold, for seconds each, with no phase weighted more than another. A third group practiced cyclic hyperventilation with breath retention, focusing on long, deep inhales followed by holding breath, effectively the reverse of cyclic sighing. Lastly, one group did nothing but mindfulness meditation, observing the breath without trying to control it, which served as the closest thing to a true control condition. Before and after each session, and throughout the month, researchers tracked mood using the positive and negative affect schedule, monitored anxiety with a standardized state anxiety inventory, and directly measured physiological arousal by recording respiratory rate and heart rate during the sessions. All 4 groups showed improvement. It's important to state clearly that paying attention to your breath, even for 5 minutes a day, has a positive effect. However, the breathwork groups outperformed the meditation group, and those practicing cyclic sighing saw the best results on 2 specific measures. They experienced the most significant reduction in resting respiratory rate and the greatest improvement in positive effect, which refers to the sense of energy, calm, and well-being, rather than just the absence of anxiety. Positive effect among breathwork participants increased by an average of 1.91 points on their scale over 28 days, compared to 1.22 points in the meditation group. The mood impact was measurable after just 1 session, and it strengthened with repetition. Notably, the deepest or most forceful breathing didn't yield the best results. Cyclic hyperventilation, focused on big inhales and breath retention, performed worse in calming outcomes. Breath patterns dominated by inhalation tended to escalate arousal in the nervous system rather than reduce it, consistent with the earlier discussion on chemoreceptors. Inhale-heavy patterns typically increase CO2 rather than stabilize it. It was the exhale, specifically, that made the difference. Spiegel, one of the study's lead authors, emphasized a key point. Breathing occupies a unique spot in the nervous system. It operates mostly automatically, like heartbeats or digestion, but can also be controlled voluntarily in ways those other functions cannot. You cannot consciously slow your gut, but you can choose your next breath. By making that choice, you tap into a control loop that influences your entire physiological state. One important detail the Stanford trial did not measure is often misrepresented in summaries. The study tracked mood, anxiety, respiratory rate, and heart rate. It did not take blood samples or measure cortisol levels. Given the biological mechanisms involved, it's believable that a practice which calms the autonomic nervous system might also, over time, affect the hypothalamic pituitary adrenal axis and the cortisol it controls. However, this specific link hasn't been directly shown in the data, and claiming it as a proven fact would exaggerate the findings. The more accurate, and possibly more intriguing, version is that a 5-minute daily breathing pattern improved self-reported mood and objectively measured respiratory rate over a month in a controlled trial, more than simply watching your breath did. This part of the story feels strange to me. A behavior with a 500 million year evolutionary history, driven by 200 identified neurons designed to reinflate small sacs in your lungs, turns out to be one of the quickest ways for a person to reduce anxiety in real time. According to the Stanford data, it works faster than meditative practices just focused on watching the breath. The same mechanism that prevents your alveoli from collapsing can also help you slow your heart rate. Now, let's clarify the evidence, since these layers exist at different levels of certainty, and it's important to know which is which. The mechanical function of a spontaneous sigh, reinstating collapsed alveoli, is well-established physiology, documented over decades of respiratory research and directly observable in patients on ventilators who lose the reflex. The Krasnow and Feldman circuit, which includes the RTN neurons, bombesin-like peptids, and prebotzinger receptor neurons, was mapped in mice and rats using direct genetic and drug manipulation. The equivalent structure is presumed but not independently confirmed in the human brainstem, although the preserved structure of the mammalian respiratory rhythm generator makes this a reasonable assumption. The effect of extended exhalation in slowing heart rate is well documented in humans through decades of heart rate variability research. The Balban, Spiegel, and Huberman trial is a genuine randomized controlled study, peer-reviewed, with a sample size of 111, good for a breathwork trial but modest by large-scale clinical medicine standards. It measured self-reported mood and standard physiological markers rather than cortisol levels or brain scans. The finding that cyclic sighing specifically surpasses other breathwork techniques in respiratory rate and positive mood is based on a single study, which is not yet widely accepted, and the field would benefit from larger-scale replication. What you should remember is not that this breathing method is medically proven to cure anxiety, but that a mechanism this concrete, traceable from a neuropeptide, to a brainstem nucleus, to a nerve, to a heart rate, to a measured mood in real people, deserves more recognition than the typical advice of just take a deep breath. One detail in the Krasnow and Feldman paper lingers with me more than the rest. Before they discovered the sighing neurons, no one had really searched for them. A sigh appears as nothing, a simple exhale, the kind of action you'd cut from a video, too ordinary to merit funding from 2 labs or years of transgenic mouse experiments. 2 separate labs on opposite coasts, Krasnow's at Stanford, focusing on the molecular side by examining neuropeptide genes, and Feldman's at UCLA, mapping the pre-Botzinger complex since proposing its existence in 1991, had to collaborate to confirm a single circuit. It took targeted removal of a few hundred cells from the approximately 86 billion neurons in a mammal's brain to show that this small, overlooked action was crucial. Removing it didn't just lower a statistic, it changed how a living animal's lungs functioned, breath by breath, for the rest of its life. Much of what keeps a body functioning is like that. Not the dramatic systems, but the quiet, 5-minute maintenance behaviors that operate beneath a lifetime of conversations, arguments, meals, and grief, never asking for acknowledgement. So, think back to the breath you took at the start. 2 inhales, with the second being small and sharp on top of the first, opening up any alveoli that had started to sag since your last deep breath. Then, the long exhale, lasting 3 or 4 times longer than the inhale, sending a slow pulse down the vagus nerve to your heart muscle, gently telling the sinoatrial node, in the only way it understands, that whatever emergency your sympathetic nervous system was preparing for is, for now, over. Somewhere beneath that, a disrupted breathing pattern was reset to its normal range, similar to how a rat immediately sighs more when a threat is removed. 200 neurons in a nucleus, you'll never feel allowed all of this to happen simultaneously. None of this relied on belief. You didn't need to feel calm first, think the right thought, or want it badly enough. It required air moved through your body in a specific order for about 30 seconds. The sigh was never just a sign of feeling better. It was the mechanism for it, happening 9 or 10 times an hour, whether you notice it or not, regardless of whether you ever read this or not. You simply took control of a system already in motion and asked it, once, on purpose, to do what it already knew how to do.